Hello everyone. Let me take you back to a time when biology was still unfolding its greatest secrets. Imagine looking at dividing cells through an old-fashioned microscope without the clarity we enjoy today. What would you see? In the late 1800s, curious scientists observed strange thread-like structures inside the nucleus, especially when cells were dividing. They didn't know what these threads were, but they suspected they had something to do with heredity, the passing down of traits from parents to children. Just like detectives looking for hidden clues, these early scientists used microscopes to peek into the secret walls of life, uncovering what we now call chromosomes. The real breakthrough came in 1882, when a German scientist named Walther Flemming became the first to study these mysterious threads in detail. He observed them in the dividing cells of salamanders, amphibians whose cells had large chromosomes that were easier to see. Flemming noticed something fascinating. During cell division, these threads seemed to split lengthwise. He described this process and gave it a name, mitosis. The word comes from the Greek word mitos, which means thread, perfectly capturing their appearance. Isn't it amazing how a single observation could open the door to modern genetics? Now, remember, Flemming did not have the powerful microscopes that we use today. His instruments were basic, yet his patience and skill made the difference. Through careful observation, he drew detailed sketches of the nucleus, showing how long, thin chromatin fibers condensed into distinct chromosomes during division. Even with limited tools, his sharp eyes and keen curiosity allowed him to see what no one else had clearly described before. As you can see in this diagram of chromatin fibers turning into chromosomes, Flemming was witnessing life's genetic blueprint being carefully packaged. You might be wondering, why did Flemming choose salamanders? Well, their chromosomes were much larger compared to other animals, making them perfect for observation. The bigger the structures, the easier it was to track their behavior under the microscope. Today, biology students use onion root tips for the same reason. Their dividing cells are clear and plentiful. Isn't it fascinating how one species can become a window into understanding processes that happen in all living things? Salamanders helped Flemming uncover something universal, the dance of chromosomes during cell division. Flemming's work marked a turning point in biology. For the first time, scientists had clear evidence that these thread-like structures inside the nucleus were not random, but organized carriers of hereditary material. His introduction of the term mitosis and his precise drawings gave biology a new direction. Later scientists confirmed that chromosomes were indeed the vehicles of inheritance, the link between one generation and the next. Because of this foundational contribution, Walther Flemming is often remembered as the father of cytogenetics, the branch of biology that studies chromosomes. So let's quickly connect what we've learned. The discovery of chromosomes began with careful observations made by Walther Flemming in 1882. By studying salamander cells, he saw thin chromatin fibers condensing into visible chromosomes, and he named their division process mitosis. His pioneering work gave us the very first evidence that chromosomes were central to heredity and cell division. And think about it. From Flemming's simple sketches with old microscopes, we've now advanced to powerful electron microscopes and molecular techniques, unraveling DNA sequences and mapping entire genomes. All of this began with the curiosity to look deeper into a tiny dividing cell. I hope you now appreciate how a single discovery, made over a century ago, continues to shape modern biology. Chromosomes truly are the storytellers of life, carrying the legacy of generations. Until next time, keep your curiosity alive, because every great discovery begins with the simple act of looking closely at the world around us.